Good evening. My name is Nicholas Hanley, and today I'm going to be talking to you about insulin and glucagon. The objectives for this presentation are number one, to explain fuel metabolism, number two, to compare and contrast the absorptive and post absorptive states, three, to distinguish the differences between the various cells that are found in the islets of Langerhans, and finally, to compare and contrast insulin and glucagon, where most of today's uh, presentation will take place. Okay, so fuel metabolism. Fuel metabolism basically defined it's the amount of nutrients in our diet that we must have in order to sufficiently meet our body's needs. Um, the needs we have are for energy reduction and cellular synthesis to keep everything operating the way it needs to in our body. What requirements do we have? And when we're looking at this fuel metabolism, there are two considerations that we're gonna look at. Number one, the nutrients from the meals must be taken in and stored and then released between meals. Okay, so we're not constantly supplying our body with energy, we're not constantly eating. So we have to have a way, a mechanism in place that allows us to still provide energy between meals. And the second major consideration is a pretty important one. Um, the brain must have a continuous supply of glucose. It needs it whether we're awake, sleeping, doesn't matter. It constantly needs that glucose supply, which is why we have to maintain a certain blood glucose level to keep everything running smoothly. Um, to accomplish these facts and reach this food fuel metabolism needs that we have, our body stores energy in three ways, or in three molecules. So with glucose, fatty acids, and amino acids. If we look at the glucose, this is our primary source of energy. We store excess in our liver and muscles as glycogen. So um, once we fill that glycogen store, any excess glucose then gets converted to fatty acids and glycerol, and we use that to make triglycerides, which is our adipose tissue, and that's our secondary storage. Um, but this glucose provides our main energy source and gets us going. Once the glucose is gone, our next, like I said, energy source will be our fatty acids. We get them generally from our dietary intake, and we incorporate them into the triglycerides, again, stored in adipose tissue. Finally, a third option for an energy source would be our amino acids, or the circulating amino acids. Um, and these ones generally aren't immediately needed for energy. These are more for protein synthesis and structural things, but the amino acids that aren't needed for protein synthesis can be converted to glucose and fatty acids and then stored as triglycerides. Um, so overall, adipose tissue is where we're going to be storing a lot of this excess um, energy that can be then released during these um, fasting times. Okay, so now if we look at the two main states, we have an absorptive state and a post-absorptive state. The absorptive state is the fed state. It's where we've been eating and we actively have food present. So in this absorptive state, um, we are absorbing the nutrients in the food into the blood. In this case, glucose is plentiful, our major source of energy, so we're very happy. At this point, we're using very little of the fats and amino acids as energy sources. And then any extra nutrients or excess nutrients not needed immediately are going to be stored away for later use. Um, Opposing that is our post-absorptive state. So this is, the times when we're not eating, so between meals. Um, it normally takes about 4 hours for the average meal to be absorbed. Um, and if you eat every 4 hours, that's fine, breakfast, lunch, dinner, but that only gives you 12 hours in a day and you have another 12 hours. So we got to find something to do with the time between those. So the post-absorptive state is our fasting state. In this case, what we're doing is we're taking those stored energy sources, the nutrients we stored in the absorptive state, and then we're breaking those down in order to use as energy sources. So gluconeogenesis, using amino acids to produce or build glucose molecules, glucose sparing, where we're using the fatty acids in place of the glucose so that the glucose that is in the system can be used by the brain. Um, so in gluconeogenesis, again, carbohydrate synthesis by other means. Um, but in the post-absorption stage, glucose as an energy source is greatly reduced. Um, but overall, between these two states, the concentration of nutrients within our blood remains relatively stable, constant, to try to keep things where we want it to be. Now, if we look at the islets of Langerhans, let's start talking about how we're going to be maintaining these levels. The pancreas is a pretty important organ. It has both endocrine and exocrine functions. And basically, we're going to focus more on the endocrine functions here for this presentation. But amongst the exocrine cells in the pancreas are clusters of endocrine cells that we call the islets of Langerhans. Within these little clusters, you have various cell types. So the first cell type are the alpha cells. These make up about 25% of the endocrine cells, and the alpha cells are responsible for producing glucagon. So we'll come and talk about them again in a little bit. Next, and the most common cell you have in the pancreas or in the islets of Langerhans are the beta cells. They make up about 60% of the cell's mass for the endocrine cells, and they produce both insulin and amylin simultaneously. So an insulin tends to be our most important pancreatic enzyme or hormone that we're going to talk more and more about. Of these two enzymes or hormones, insulin is produced 100 to 1 compared to amylin, so insulin's greatly more in production. Next, you have your delta cells, which would be about 10% of the endocrine cells. And this is the pancreatic site of somatostatin synthesis, which is a topic for another lesson. Then we have our gamma cells. They're only about 4%, and these secrete pancreatic polypeptide, and they play a role in reducing appetite and food intake. There's not a whole lot understood about these, or they're not very understood right now, so we'll get back to them. And finally, you have your epsilon cells, which are less than 1% of the total endocrine cell mass. These are very newly found, relatively speaking, and they've been found to release the hormone ghrelin, and this is sometimes called the hunger hormone. And basically, it's mostly secreted by the stomach before eating meals. But of these, your beta cells tend to be on the interior or centrally in the islets, and the other cells tend to be on the periphery, moving around them. So again, this presentation is more about glucagon and insulin, so we're going to focus in on those now. So with insulin's main job is it lowers blood sugar or blood glucose levels, and that's what most people think of it as. But it also helps reducing or lowering fatty acids and amino acid levels through other means. It is the only hormone that actually reduces blood glucose. So insulin is going to promote cellular uptake of these hormones or of these nutrients and help convert between glycogen, triglycerides, and proteins in these building blocks here. It facilitates glucose transport into the cells. It's also going to stimulate glycogenesis, or the building of glycogen, and it's going to inhibit glycogenolysis and gluconeogenesis. Glycogenolysis is where we break down triglycerides to build glucose molecules, and gluconeogenesis is where we take amino acids and build glucose out of them. And both of those happen in the liver, but their job is to increase blood glucose levels. So insulin generally works to reduce it. So stimulating glycogenesis, where you're building glycogen from glucose and importing glucose into the cells, reduces blood glucose levels. And by inhibiting the other two, glycogenolysis and gluconeogenesis, you also help to reduce the glucose level in the blood. Now, glucose generally cannot just cross through the cell membrane. It generally requires a glucose transporter or glut transporter, and it goes through facilitated diffusion. So it does move through the concentration gradient, but it needs the glucose transporter to allow this to happen. There are 14 different forms of the glucose transporters. I'm just going to point out a couple of them as we go through this. GLUT1 is essential for the brain, and it's non-insulin dependent. And most other cells, especially muscle cells and other body cells, have a GLUT4 container or GLUT4 transporter, which is insulin dependent. So on these ones, as insulin connects to its insulin receptor, a signal is sent for a vesicle with a GLUT4 transporter in it to move to the cell membrane or plasma membrane. As the vesicle fuses with that, it inserts that transporter into the plasma membrane, and then it allows that transport of glucose to occur. These GLUT4 transporters can also be induced to fuse with the membrane via exercise. So that's going to be important when we talk about the different types of diabetes here in a little bit and some of the treatments for them. So this GLUT4, again, is in most resting muscle cells and other body cells. Working muscle cells and liver cells tend not to need it as much because we can transport it there throughout that. This excitatory and secretion coupling process. So how do they actually go through and secrete insulin? As we're looking at this, glucose is going to enter these cells through the beta cells via GLUT2 transporters. So this allows glucose to go in. As soon as that glucose passes through that, it gets phosphorylated into glucose 6-phosphate, which is then oxidized by the beta cells to yield ATP. Okay, now there are two types of channels that are important in this whole process. There's an ATP sensitive potassium channel and then a voltage gating calcium ion channel. Once this ATP is generated through the phosphorylation and oxidation of the glucose 6-phosphate, that ATP can then go and act on the AP-sensitive potassium channel, which is normally open and allows potassium ions to leak out of the cell. In this case, once ATP binds, it closes that gate and prevents the potassium from being able to leak out, which causes the membrane to depolarize. So because of the less outward movement of the potassium ions, that depolarization will then activate the voltage gated calcium channels. Okay, and they're normally closed at rest, so when they get activated, they open up there, allows calcium to rush into the cell, and this calcium triggers the secretory vesicles containing the insulin molecules to go and fuse with the cell membrane and release the insulin out. So a pretty important process. This can also insulin can also be secreted through the action of elevated amino acid levels. Again, what we're trying to do here, increased insulin enhances the entry of amino acids into cells, and that lowers their levels in the blood, and that promotes protein synthesis. And then we do this by increasing or secreting, by generating ATP, which leads to this excitation secretory coupling. Insulin can also be triggered to be released by gastrointestinal hormones that are secreted in response to food intake. Some of those are glucose-dependent insulinotropic peptide, or GIP, and glucagon-like peptide 1, or GLP-1. Again, they stimulate the insulin secretion kind of in an anticipatory fashion. So before you're really completely eating them, they're triggering that to happen. And these are called incretins. which are basically hormones released from the digestive tract that help notify beta cells of impending rise in blood nutrients. So again, that anticipatory factor, and these work by increasing the CMP or CAMP, which enhances the calcium-induced release of insulin. And then the autonomous nervous system can both parasympathetically and sympathetically trigger release of insulin. And again, this is more in response to fight or flight or stress exercise than it is to diet or blood glucose levels. Now let's look at glucagon. With glucagon, it's kind of the opposite of insulin. What we're doing here is these are going to be secreted by the alpha cells of the pancreas, and this is mainly acting through the liver because we're going to take that glycogen we built up and break it down into glucose and then release that glucose back out. So that's the main thing there. For carbohydrates, this increases hepatic glucose production, okay, and that's what we just talked about, the glycogenolysis, where we're breaking down glycogen and producing insulin. This can produce a hyperglycemic effect, and it's going to decrease glycogenesis and promote both glycogenolysis and stimulating gluconeogenesis as well. So all of these things kind of happen together, and glucagon is kind of the instigator there. It can also promote lipolysis and inhibit triglyceride synthesis. So instead of building triglycerides, we're starting to tear them down and use them to build more glucose. And this can increase ketone production and convert fatty acids to ketone bodies. And then for proteins, we're going to inhibit hepatic protein synthesis here, and that we're going to promote the degradation or breakdown of these hepatic proteins. By doing this, gluconeogenesis can further be contributed to the glucogen catabolic effect on hepatic protein metabolism. Either way, what we're trying to do is just produce more glucose into our bloodstream. So if we look at the two, they have a direct negative relationship with blood glucose levels, which means I always like to tell my students, when you see the little figure 8, they want to maintain the levels in here. One of them, for insulin, if the blood glucose level goes up, insulin's going to act to bring it back down within that normal range. If blood glucose levels go down, glucagon's job is to bring it back up to that normal range. So they both have direct negative relationships, but they work in opposite directions. So for this, they work as a team to maintain blood glucose and fatty acid levels and direct that negative feedback relationship between blood glucose levels. And both beta cells and the alpha cells rate of secretion, but again in opposite directions. So if blood glucose level increases, the production of insulin is going to increase, but the production of glucagon is going to decrease because we want to do those things that are going to limit the amount of glucose in our blood and not encourage it. Whereas if the blood glucose level is depressed, then the insulin production is going to be depressed and glucagon production is going to be elevated. So again, we're trying to keep that homeostatic level of where we want to be. For fatty acids, it's the pattern or behavior is similar to that of the blood glucose. So if we have decreased levels of fatty acids, we're going to see decreased levels of insulin and increased levels of glucagon. So if there's less fatty acids, we're going to stop absorbing them and we're going to start releasing them by breaking down the stored energy we have. Amino acids tend to have a little more of a unique behavior or unique relationship. If there's elevated amino acids, we actually increase the production of both insulin and glucagon. For example, this would be seen like if you eat a meal that's high in protein and very low in carbs or no carbs at all. In this case, if you've had that, your your body's going to be triggered, the insulin is going to be produced, and if we do that, the glucose level is going to go, or the glucose is going to move into cells, reducing the blood glucose level. Um, if we didn't also release glucagon, then we wouldn't be replacing that glucose in the blood, and that could cause hypoglycemia or low blood sugar levels, and it could cause the brain to lack that supply that it constantly needs. So we've got to be careful on those things, but insulin and glucagon does work in that factor. Its main thing is blood glucose level, and we trigger different things to happen depending on where that blood glucose level is. Some disorders that were discussed within this that kind of to mimic things. Um, Diabetes mellitus is the most common endocrine disorder. In this condition, um, you have problems with insulin. Either it's not produced at all, as you see in type 1 diabetics, or there's such reduced sensitivity because of overexposure to the insulin or the high blood sugar levels that you see in a type 2 diabetic. There's both of these end up in elevated blood glucose levels or hyperglycemia. Um, they have large urine volumes, constantly going to the bathroom. We could see things like glucosuria or glucose in the urine, um, polyuria, which is excessive urination. Um, because you're excessively urinated, that could kind of, as you're pulling that sugar out, you're also pulling water with it, and that can lead to dehydration. When you're dehydrated, the blood is a little bit thicker, so it's harder to pump. So you end up with circulatory problems, and could eventually or lead to death if those problems aren't solved. Could also cause renal failure because of the inadequate filtration pressure. Um, and losing water can cause the cells to shrink. So there's brain cell shrinkage that can occur in other nervous and other cells. And then polydipsia and polyphagia. Polydipsia being excessive thirst. If you're dehydrated, it's a natural mechanism to drink more water. And polyphagia, even though you have a bunch of sugar, your cells aren't getting it. So you might have that feeling that you're always hungry. So you're just kind of exacerbating the situation by providing more sugar into the mechanism. It can also, if we're elevating these levels, um, fatty acids can release excessive ketone bodies because we're not stopping lipolysis and all those things from occurring. So we can get ketosis, and because we have acetoacetic acid building up and all these, we end up with metabolic acidosis, which can depress brain function and leave us with fruity smelling breath and things like that. If we're consequence of protein metabolism here, if we're breaking those down because we can't get the glucose, we could lead to muscle wasting and weakness due to breakdown of muscle proteins, and that would cause weight loss and reduction of overall growth in children. So excess amino acids in the blood means reduced uptake and increased degradation, and it can be, those amino acids are normally used for gluconeogenesis, which will further produce more glucose, exacerbating the situation as well. And then we have with diabetes mellitus, sorry, I want to go back to that. What they've found is some of the new research out there is instead of just treating with insulin, combination therapy with both insulin and glucagon is showing some promise in having better control over maintaining the blood glucose levels in the range we want it to be. And then hypoglycemia is where you have low blood sugar. Now, this can result from either a type 1 diabetic injecting too much insulin at once or having oversensitive production of insulin, too much produced naturally in your body. What happens here is that affects the brain's glucose supply. If your insulin makes the glucose go into the cells, which lowers the blood glucose level, leaving the less glucose available for the brain. And then excess glucagon can aggravate this situation as well, because if we're going through and we're um, normally in glucagon, insulin is required for glucose to gain entry into the cell, into the alpha cells, where it can control the glucagon secretion. So if we're not having insulin there, if it's not reacting properly, um, we're not going in. We can't inhibit. I'm sorry, that results in hyperglycemia. I'm looking at the wrong thing here. Um, Hypoglycemia, low blood sugar, too much insulin, and then excessive glucagon can aggravate that, because if glucagon's job is to go through and trigger more things to happen. Um, so if we look at our objectives again, our goal was to explain fuel metabolism. Number two, we wanted to compare and contrast the absorptive and post-absorptive states. Next, we wanted to distinguish the difference between the various cells in the islet of Langerhans, and then compare and contrast insulin and glucagon. I think we did a fairly good job at that, and here are my references, and thank you for listening.